This is the first time I'm actually referred to as Dr. Young for a talk, so this is really exciting. Thank you everybody for joining on this Twitch stream. I'm very excited to be a part of this exciting and enterprising opportunity. Dr. Meekins is doing a fantastic job with this community, and just to be able to contribute a little bit of some science that I'm doing here, I'm very excited. So basically today we're going to talk about the twists and turns of DNA and the importance of DNA deformability and the local features that influence the way that the genomic structure and function relationship occurs. I ask that if you have any questions, just let us know in the chat room and whatnot. So let's go ahead and just begin. I've always been fascinated by higher ordered structures of DNA and how they have this similarity to a Tokyo subway station at rush hour. It has this feel of a centralized hub of organized chaos. From a physics perspective, I'm amazed at how these compact structures maintain an essential balance between protecting genetic material and granting access for expression. These condensed structures of compact DNA contain a mix of RNA and proteins used as machinery for structure and for the expression of genes as needed, and are highly dynamic, influenced by local level features. The higher ordered chromosomes are made up of repeated subunits, either in the form of supercoiled loops or plectinemes, or a regular fiber, chromatin, in eukaryotic cells. These protein-mediated constructs locally deform DNA at one or more sites, or wraps around the macromolecule around a protein complex in a sort of nucleosomal subunit. These supercoiled loops and chromatin fibers may be modified to change how the DNA strands are with something known as topoisomerase enzymes. However, just as sequence plays a role in expressing genetic material, so too does structure. Certain proteins are more likely to bind to tracks of DNA based only on their physical states more than sequence. And for some people who study genetics, we've always been taught, sequence equals, function and sequence equals binding, but that's not always the case. These binding interactions will locally distort DNA, causing additional stress along the polymer, either upstream or downstream from the binding site, causing additional stress that must be compensated, which in turn causes more. So it kind of has this cascading effect on the level of deformation along the local regions of DNA. So developing computational models of DNA at this mesoscale level leads to insights in the relationship between local features and global structure. I'm interested in understanding how binding of proteins and small molecules may enhance or repress DNA function. I'm also interested in determining features that make certain configurations more biologically favorable, such as nucleotide length or sequence composition. So for my talk, I'm going to break this up into three different components here. I first want to talk a little bit about our theoretical framework and how we can develop models of DNA. I will then discuss a project that was conducted by in our lab from a paper that we had published in 2021 that had discussed developing mini-chromosomal circles of DNA and how nucleosomal variation influences the free non-bound DNA. And finally, I'm going to talk about a recent study that updates a knowledge-based sequence-dependent potential that allows us to expand what we know on DNA modeling. When we go through and develop our computational models, it's important that we have a grasp of how best to model our DNA. We use a program known as emDNA, and the use of emDNA requires a shift in how we can describe a collection of DNA away from XYZ Cartesian coordinates. For example, a collection of 100 base pairs can result in well over 3,000 atomic coordinates, let alone all the records that you would need for all the atomic relationships. This is a lot of information that needs to be stored and used regularly. However, if you take a closer look at this structure of B-form DNA, you might notice that there is a regularity to this. There looks like there's almost a planar consistency to our structure, to the point where we can place rectangular frames over these base pairs. And by doing so, we can collect global positions of the base pair origin and its local three-dimensional configuration in space. And we can do this because deformed DNA is the more biologically active form of DNA, and it just happens to be nicely regular. From this collection of the reference frames that we can generate a series of 6x1 vectors of these base pair step parameters. Each vector contains 3 rotational parameters known as tilt, roll, and twist, and then 3 translational parameters known as shift, slide, and rise. Each similarly generic, we can still impart sequence specificity that highlights the sequence deformation along these parameters for our 16 different dimer sequences, of which 10 are unique. And I mentioned this program that we use called emDNA. emDNA is an in-house software that was developed by a colleague of ours, Dr. Nucleosomal, and I butcher his name all the time. I'm a North Carolinian. I'm not really great with French, and I apologize profusely for that. He always makes fun of me for it. We use this software known as emDNA that allows us to take a model of DNA and elastically optimize it. And what's really nice is that the workflow for emDNA is rather simplistic. We can use step parameters that we've talked about with those vectors I mentioned earlier, and we minimize elastic energy for that fragment of DNA. We can see the general workflow here. What's nice is that emDNA computationally is its basic usage. All we really need is one file in order for the optimization to occur, either in reference frame formats or step parameter formats based on a scheme known as 3DNA. So there's a website known as Web3DNA, which you're free to check out as you wish, but it's a format that is either base pair step parameters or reference frames. All we need is one file that has that information. There are certain requirements that must be selected when we run emDNA, such as what we want our model rest state to look like, or our force field, but this is not to be confused with molecular dynamics force fields. What we refer to as a force field consists of a rest state of a collection of step parameters representing some zero energy configuration, and then a 6x6 elastic stiffness constant matrix representative of some XY sequence step. This matrix here contains coupling data from each of the 6 parameters. So the rest state parameters, as well as the elastic stiffness constants, are also in the tilt, roll, twist, shift, slide, rise space. Rest state models can range from a simplified model, which we refer to as ideal DNA, which has no sequence dependence. All the step parameters are identical. Changes to the stiffness are identical, to a fully sequence-dependent model that has different step parameters and elastic constants for each dimer that we use. These models are built into emDNA along with this MAT packager tool that allows for custom rest state models. emDNA also includes a novel feature that allows us to differentiate from models like molecular dynamics or Monte Carlo. It allows us to apply constraints to our system. We can freeze ends of the DNA fragments in a specific position and or orientation in space. Not only that, we can also specify different regions along our fragment and, quote, freeze them out of optimization. So an example of this would be if I was to have this structure, the circular structure here that's rotating on the top, where there are 4 specific U-bends within there. For those of you who are colorblind, I apologize, but hopefully you'll notice that there's a difference in tint where I have green, magenta, blue, and yellow coloring to denote these 4 U-bends. What I can do is I can determine elastically optimized configurations of this circular structure with some or all of these U-bend states held fixed. Once optimization is complete, we can use additional tools like 3DNA or PyMol, the Python modeling software that allows us to develop more robust 3-dimensional models oriented along the principal axes, highlighting differences in configurations based on, for example, if we want to free DNA between sites 1 and 2, yellow and blue, sites 1 and 3, yellow and green, and sites 1 and 4, yellow and magenta. And for each, we have two different views along the principal component axes. And you'll notice that there are different energy values. The lower the energy, the more biologically stable and relevant the structure is. So something that is linear, that forms a linear overall structure, is lower in energy because our rest state was modeled as a linear model. Now, getting the actual optimization requires us to have some level of initial condition. And developing initial conditions, we can absolutely do a bunch of different things for. We can make a simple DNA mini circle with the sines and cosines functions that you can see right here to get the tilt and roll values with respect to a specified length and twist. More rigorous mathematical techniques can be used, such as anchoring a Bezier curve to two different base pairs of a known position and orientation. There is an additional tool in emDNA that allows for a ramping of a specified configuration, like some superhelical nucleosome, into a target fragment seen in this video. This linear ramping procedure iteratively alters the selected regions in gold to model the target fragment. And once we do that for every iteration, the remaining free DNA is optimized. So our goal is to basically take a circle and put in a nucleosome and get a resulted structure. And that now becomes an initial condition for more robust studies. So here, for this section, I want to talk a little bit about the study I mentioned earlier with my colleague, Dr. Stefir Todoli, who also came from our lab. And we focused on the structure of the nucleosome. We used a data set of well over 150 high-resolution crystal structures found from the protein database. And we were able to observe variations along this wrapped DNA complex that it influences the overall structure of the adjacent DNA. And you can find this article in the Journal of Molecular Biology under Robert Young and Dr. Stefir Todoli in 2021. Now, what we are seeing here is a model example of what's occurring in a eukaryotic, one particular eukaryotic virus. The nucleosome subunit found in chromatin fibers consists of the wrapped core particle and a length of free linking DNA that connects successive nucleosomes. Not only do eukaryotic cells have them, as I mentioned, eukaryotic viruses like SV40 utilizes a nucleosome-bearing model. Now, previous experimentation from the Ariel Purnell lab had actually showed that there are differences in topology along the SV40 molecule and the SV40 genomic setup, showing how many circles of various levels of supercoiling can uptake a nucleosome, as seen here in this electromicrogram of structures that have a -2 topoisomer state. Depending on the length of the circle and the level of supercoiling, either one or two nucleosomes can be uptaken by the circle. And we can do a more zoomed in view here. These mini-chromosomes show a mix of an open and closed configurational state. We can explore properties along the nucleosome regions to find the binding interactions and how they influence the global writhe of the mini-chromosomes, and understanding how certain lengths prefer different topoisomers, such as how a length of 359 base pairs favors a -1 state. So a survey of nucleosome labeled models from the protein data bank was conducted by collecting 164 high resolution structures that was analyzed by my colleague. Here we see a nucleosome core particle with its characteristic left-handed configuration wrapping about an octameric histone protein complex, and it's wrapped about 1 3/4 of superhelical turns. The DNA pathway consists of an entry and an exit gyre meeting at this central dyad base pair. A smooth B-spline curve of 141 base pair tracks for each model are shown here on the right, made up from the base pair origins and overlapping at this central dyad. This image suggests a rather dynamic structure with variation along the gyre paths and the entry and exit positions. Closer inspection of 3 particular examples overlapped at the dyad shows additional differences at the entry and exit base pairs, showing variation along the frame's orientation as well as its position. Overlapping these B-spline curves at the entry base pair position rather than the dyad shows a different perspective of the pathway variety, with two clusters based on the pathway and the end positions. Analysis of the nucleosome tracks revealed differences in the uptake of twist between neighboring base pairs, with the net twist uptake used as a color coding for each of the nucleosome paths. Additional cluster analysis was used on the 164 base pairs, revealing 7 NCP or nucleosome core particle groupings, three of which are highlighted here. Each of these groups are based on differences in the net twist uptake along the chain and twist differences at specific positions along the DNA chain, mainly at positions plus minus 50 base pairs away from the dyad and plus minus 20 base pairs relative to the dyad. These three groups in particular, which, I just went ahead far too much, sorry. Come on. There we go. Sorry about that, everyone. Additional twist cluster analysis was used on the nucleosomes, revealing 7 unique nucleosome core particle groupings, 3 of which that we're highlighting here. Each of these groups are based on differences in the net twist uptake along the entire chain, and then the twist differences I mentioned earlier. These three groups in particular, group 3, group 5, and group 6, represent groups of no excess twist, under-twisted, or over-twisted respectfully, and are highlighted here in the full fixed end collection, with group 5 nucleosome core particles occupying that smaller angular grouping that we see here in the middle. So for our modeling of the mini-chromosomes, we generated mini circles of 159 base pairs long, similar to the physical experiment from the Purnell lab, and we're having them bearing a 141 base pair nucleosome. These models were generated with a 4-point Bezier curve, which means that we use the entry and exit base pairs of the nucleosome as an anchor point for the curve, and we know that the directionality of the entry-exit base pair needed for us to indicate the other 2 points here listed at the top here in this image. Once the curves were constructed, base pair frames were added along the curve, allowing us to actually generate multiple topoisomers varying by internal twist for every nucleosome core particle. All optimizations were conducted using an ideal DNA rest state, which means there's no sequence dependence, everything's essentially uniform, and it's meant to be modeled after a linear piece of DNA. Now, in order for us to preserve any level of supercoiling that we've imparted on the initial condition, we also need to include some level of electrostatic, because without the electrostatics, based on the calculation we run, since it's all in base pair step space, it's not going to instinctively know not to allow a crossing over of the strand. So we need to impart electrostatics to ensure that the base pairs don't phase through or overlap through each other during the optimization process. Without this, the strands will intersect and the optimized states will occupy the lowest possible energy state regardless of the initial topoisomer state. So here we see a collection of optimized topoisomers bearing the same nucleosome example in gray. The red model is that of the relaxed circular state with a linking number of 34, which in this case, a change in linking number of 0. Yet the proper relaxed linking number 0 is 34.2. So that means that there's going to be some difference between ideal relaxed and the calculated relaxed. The differences in twist along the initial loop resulted in differences in optimized writhe, leading to helical strand crossings that you see along the plane. The writhing of the loop regions appears greater with the more supercoiled structures that we see here on the right, although the greatest writhe value is actually associated with the -3 state seen in blue. This is due to the negative writhe that's already imparted on the structure by the nucleosome region. We also found that the lowest energetic state is associated with the -1 topoisomer and not 0. What's great about that is that actually agreed with the experimental observations that were conducted decades ago. So we found from our own optimization calculations that based on this level of constraint and size, our computation matches what was observed for experiment, and that's for all the nucleosome core particles that we imparted into this modeling. There we go. We can organize the energetic data of all of the topoisomers as a function of a turning parameter referred to as gamma. This gamma is the angle that we calculate between the plane of the free loop and the normal vector of the mid frame. So it's the turning parameter tells us how turned around at some axis the free loop is relative to the anchoring points. We see from this scatter point, the scatter plot, the states of the greatest rise and the number of strand crossings are all higher in energy. And that topoisomers of 0, -2, and -1 have somewhat similar energies on the bottom, although they have different turning parameter angles. Interestingly, there appears to be a level of subgrouping within each energetic cluster. Further analysis of the -1 state group separated by twist clustering shows this subgrouping with respect to energy and turning angle. We see that with the change from undertwisted group 5 to overtwisted group 6 nucleosome models comes a decrease in the elastic energy and a turning parameter angle closer to 90 degrees, highlighted here in this progression of our collection of images seen here on the right. We can see example models from groups 3, 5, and 6 listed here on this -1 topoisomer state, with a mix of open to slightly closed configurations. The closed loops have a strand crossing closer to the loop apex and an indicator that the turning angle is closer to 90 degrees. Any state that is the lowest energy among the collection of topoisomers will have a higher probability of formation. Thus, the -1 topoisomers are more likely to form biologically. Yet, a closer look at the probability of states shows that the undertwisted -2 and relaxed 0 topoisomers have likelihood of about 25%, even if the -2 state has a closed configuration. In addition, the no excess twist group in purple has a possible 5% likelihood. If we recall, those group 3 nucleosomes were all undertwisted, which is very interesting to us. So in addition to these built-in models where we have a constrained 141 base pair nucleosome, we can observe differences in loop likelihood with respect to nucleosome groups that lead to a set of optimizations that we can use to observe nucleosomal breathing. Now, this effect is where the bound regions of the nucleosome peel away from a binding orientation with the histone proteins due to a weaker interaction. And this was going to occur more so at the first 2 turns of the entry and the last 2 turns of the exit gyres. What you can see as we loop over these videos, you'll see a series of optimizations where we incrementally peel off the base pairs from the frozen 141 base pair tracks, allowing them optimize with the free loop. These videos show optimized structures during the symmetric peeling of 3 selected mini-chromosomes. As the breathing continues along the gyres, loops turn from a relatively closed to open position, reducing gamma to about 20 degrees. As these movies loop, I would like to draw your attention to the behavior here on the far right in the orange. As the first 10 base pairs are peeled off either gyre, there appears to be a large resistance to the overall opening of the loop. This resistance appears to be a physical shifting of the loop apex rather than some turning that we see in the group 3 and group 5 example. As more base pairs were peeled off from the constrained region, 2 mini-chromosomes exhibited an ease in opening, leading to a rise becoming less negative and having a lower elastic energy. The over-twisted mini-chromosomal model showed a resistance to turning and thus no changes to rise in elastic energy. This suggests that for over-twisted nucleosomes, the -1 topoisomer may not actually be the most favorable upon unwrapping. If we look to this figure here of the average linking number of each topoisomer upon breathing, we do see that that resistance to turning in the over-twisted state is actually accompanying a change in the probability of the topoisomer, such that we're starting to see a 50-50 mix of equally likely -1 and 0 state topoisomers after we peel 10 base pairs away. So we were really excited about the findings here because not only did that show that the programming that we did for the emDNA software, it actually allowed us to verify what we see from experiment, and yet it allows us to go in a more robust direction if we wanted to incorporate things like sequence. Seeing as this is mini-chromosomal and chromosome anchoring doesn't really have sequence favorability, the question of what kind of sequences would work here is an exciting direction that we want to go in. And not only that, many chromosomal circles don't have to be just one anchoring with one nucleosome. We've seen that you could have 2 nucleosomes added to it, which adds another set of headaches. Another set of experiments that we can conduct. I use headaches in the funny sense. I'm trying to be funny. I apologize. All right. I'm having an issue transitioning, so I apologize. So up until this point, our molecular models were generated from a rest state that did not account for sequence. They all were ideal rest states that have no sequence bearing to them. These ideal models were still useful. They still allowed us to see and explain what we saw from experiment. But there is a lot of information that can be found when considering sequence-specific effects. So here I will discuss a very recent project in which we developed an updated knowledge-based potential with elastic constants. This update, which we just had submitted and published in the MDPI journal Life, and I think it was like late May, a series of submissions, includes a shift in how we can conduct our optimization. I mentioned previously that the organization of DNA is protein mediated. This is not always the case, as circular forms of DNA have become more observed in eukaryotic biology with various levels of supercoiling. These extrachromosomal circular DNA fragments are often derived from viral genomes or derived from repetitive sequences. However, a recent study of about 200 to 400 base pair microDNAs in embryonic tissue was found to contain sequences that map to specific genes in its chromosome. The organization of mitochondrial DNA is also circular, made up of a network of large maxi circles interlocking with more smaller mini circles. And in recent surveys of cancer cells, we're starting to observe more with better quality, smaller forms of circular DNA. An example of circular DNA that got my attention early on in my research was the work that was conducted by Drs. Lynn Zetrick and Dr. Jonathan Fogg in collaboration with Dr. Sarah Harris out of the University of Leeds. And Dr. Zetrick and Fogg are out of the Baylor College of Medicine in Texas. A 2015 paper showcased cryo-electron tomography maps and molecular dynamics trajectories of a 336 base pair sequence developed as a means for targeted gene therapy delivery. Now, these mini circles are called mini vectors and have a vast diversity of supercoiled states, with each topoisomer exhibiting one or more unique configurations. Now, these mini circles contain 180 base pair lambda integrase ATTR sequences, a byproduct of this site-specific recombination seen here. The ATTR site seen below here in the bold and underlined sequence is considered to be highly curved. The paper that I discussed reported a vast array of topoisomers with discussion on local deformation, with regions of high dimer bend or of low step twist. More recent publications highlight localized regions of activity with a BAL31 nuclease, which is an enzyme with no solved structure, whose functions are hypothetically dictated by the presence of exposed unpaired base pairs. More negative topoisomers were reported with high reaction rates in the presence of BAL31. This map here on the top right shows the locations of the 3 sites in particular, either in the curved ATTR region in the gray and in the blue, or downstream from the starting point, and the colored sites along the coding sequence match the map here. There we go. To fully investigate this activity, a sequence-dependent model is going to be required. Now, with advancements in biophysical experimentation and characterization resulting in more high-resolution structures in the protein data bank, an update was conducted from a previous survey conducted in 1998. This was done first by collecting X-ray crystal structure data from the protein data bank, looking specifically at protein DNA interaction data from the PDB, courtesy of a colleague of ours, Dr. Luke Zoppla. Analysis of the collected deposits show an almost exponential increase to dimer sequence data over the last 20 years, with the exception of the pandemic-related leveling off that we see on the top. In addition, we see a lot more high-quality data, with no less than 2,000 entries for each of the 10 dimers at resolution 3 angstroms or better. This increase in data supports a shift in how we can describe the dimer step. Previous experimentation and computational studies have discussed how base pairs in the context of their neighbors influence their structure in responses to stimuli. Certain sequence tracks are more susceptible to these changes that lead to a large-scale deformation, such as we see here by interlocking A form and B form DNA, as we can see for this highly curved structure, compared to a strand of B form linear DNA. Data from a previous survey of the PDB provided insight into the 10 unique dimers, such as step deformation with respect to volume of the step parameter space. Now, this data was taken only from 92 crystal structures deposited before 1998, and the analysis suggested high deformation of CA and CG steps. Yet we now have enough reliable data to explore the influence nearest neighbors has on the internal dimer step and how they factor into the configuration of the overall DNA structure, as seen here with the generic MN step in the context of either X or Y neighbors. The sequence diagram on the right highlights all possible 256 tetramer sequences, and the bidirectionality of DNA reduces that to 136 unique tetramer sequences, seen in black here. The object of this study centers on the development of the new knowledge-based potential. Analysis of data collected from over 3,000 high-resolution crystal structures was conducted to determine both a dimeric and a tetrameric elastic potential. We explicitly chose to work with X-ray crystal structure data rather than include cryo-EM data and solution NMR data. The figure on the left shows counts of the dimers and tetramers after 3 sigma analysis culling of the entire data set, with over 5,000 instances per dimer and over 160 instances per tetramer, which leads to a robustness of a data set exceeding that from the 1998 survey. And further refinement to 3 angstroms or better resolution provided a data set which we consider to have high confidence in. Once refined, we then use these models on those 336 base pair topoisomers we mentioned earlier, whose initial states will be just devised from a simple planar circle with various levels of uniform twist. Analysis of the updated model shows how the dimers in the context of the tetramer vary, in this case highlighting base pair step twist. The lower triangle here contains dimer-specific values with over-twisted dimers of NA steps and an under-twisted AT step. The larger tetrameric triangle shows very similar trends in twist to the dimer, with a few tetrameric steps that stand out. The TAAG tetramer at the top left shows under-twisting in an over-twisted section. We also see extremes of twist in the under-twisted CATA step and the over-twisted ATAG tetramer step. This trend in twisting is also seen in deformability. taken from the volume in the step parameter space for each step. We see a stiffness in the purine pyrimidine or RY steps, regions of undertwisting, and great flexibility among the overtwisted purine pyrimidine or YR steps, with notable exceptions of the relatively stiff TCAG and GCAT steps. Evidence of this nearest neighbor influence can be seen between the TGGT and the CGT steps here at the intersection of the blue and green table here. So we see this right there in the middle. There's this massive change, even though the internal dimer is the same, where a replacement of the thymine residues to either guanine or cytosine results in highly deformed GG internal steps. It is interesting when comparing this to the deformability data from the 1998 dimer model, which reported higher deformability among CA and CG dimers and this stiffer TA step that we see now. So, of course, we're going to expect some level of variation because our data set is more robust than what was seen previously over 2 decades ago. This updated knowledge base potential was applied to the 336 base pair topoisomers, which provided some insight into some observed nuclease activity. This figure here shows differences between the optimized and rest state twist along the mini circles for the -1, the 0, and +1 the topological states. The -1 state, a more relaxed and open configuration, was reported to have low reaction rate in the presence of BAL31 nuclease. While we see variability in the twist of the ATTR region highlighted by this black bar, we do see larger changes in both site 1 and site 3. These spikes that we see in these sites are found at 2 TA tetramers, the CTAT step in the -1 state and the TTAC step in the +1 state at the start of site 3. These steps are higher in elastic energy, leading to an increased deformable state. We can see that from the molecular models that while the -1 to +1 topoisomers do not exhibit high degrees of supercoiling, which does in fact drive nuclease activity, we do see relative positions between the 3 sites shift along with differences in the rive. When comparing the 3 knowledge-based potentials based on the sequence alone and not the initial state, it is clear that expanding to a tetrameric context is incredibly useful in developing a realistic 3-dimensional model. This central figure shows the same differences in twist as before, but with the same -1 topoisomer using each of the different sequence-dependent models. These are instances here that highlight how dimer models alone appear to miss the details that influence deformability, particularly in site 2 in blue. We see this with the TA dimer steps, which have regular overtwisting and high deformability. This site 2 slice has high TA and AT content. These are differences of about -3 degrees in the repeated TA at the start of the site, which the dimer model shows to be distorted. But this is also a TA-TA tetramer, an AT dimer in the context of a T and an A neighbor, which is undertwisted and does not appear to be deformed using the tetramer model. These differences with and without tetrameric context are highlighted in linear models of the sequence. The previous dimer model seen here shows local regions of bend, but we still get an overall linear construct. This changes greatly when we apply the updated dimer model, which shows significant curvature specifically along the ATTR region between the green site 3 and the blue site 2. And yet, when we apply the tetrameric model, we get an even more curved linear structure, supporting the evidence that it is easier with this sequence to cyclize and become a circle biologically. So upon summarization of all the work that we talked about here, the optimization of the step parameter space that we use with our emDNA software offers a new method in constructing loops and mini circles for protein-decorated DNA, rather than deal with trajectory models and focusing in on the influence of local deformations and local changes to the curvature, what we're really interested in more so is if we can constrain local regions, what would be the most elastically optimized global configuration of a structure, allowing us to influence constraint, which you don't get that level of control with Monte Carlo or with molecular dynamics simulations. And we can still offer and impart new levels of topology so we can maintain levels of supercoiling as we wish. Optimization of the 359 base pair mini-chromosomal circles confirms the dominance that we saw that was seen from experiment decades ago of the -1 topoisomer. Now, differences in twist along the bound DNA path influences the higher ordered structures of the mini chromosomes. And yet the question still remains, is that change to the twist along the DNA nucleosome, is it conditional of the way in which the biophysical experimentations were conducted? Is there a level of flexing or breathing or changing that occurs in the nucleosome so that possibly the more dynamic picture is that the nucleosome can flex and change and the differences in twists? It would be exciting to see how we can impart these differences in our elastic optimizations. And finally, expanding to a tetrameric context enhances the DNA models that we're using, capturing localized deformations found at sites of enzymatic activity specifically associated with 336 base pair mini vectors. And as we continue to work through the data with these circles, we can impart new supercoiled structures. And a lot of the analysis that, or a lot of the optimized structures that we're seeing with these highly curved structures are really exciting. And it seems as though the sequence, based on where the crossing intersections are occurring, it seems that the sequence matters. Some sequences like to be closer to that crossing than other sequences based on our model. And I'm really excited about delving deeper into that as I move forward. Thank you all very much for your time and your attention. And I'm really excited to hear about some of the questions you all might have. This was probably a lot of information. I might have gone a little bit too robust in the data. I want to take the time to thank members of my lab, especially Dr. Olson, as well as collaborators that I had mentioned throughout the talk. And funding was made possible through the NIH, as well as through Rutgers emergency funding for COVID-19. And we utilize a lot of the optimizations we utilize through supercomputing cluster with the Rutgers University Advanced Research Computing Network. So if you have any questions, now's a great time to ask. And and feel free, if you have any other additional questions after the talk, feel free to get my email address from Dr. Meekins, and I'm happy to answer whatever I can. So thank you very much for your time.
Fantastic talk. I mean, honestly, going deep into the data is not a problem. It's always glad to kind of nerd out about stuff. So,
oh yeah,
that's
it's really exciting when you consider all the options that you can do computationally.
Mm-hmm.
Because when it comes down to biologics, when it comes down to like proteomics, some of the buzzwords like with machine learning and molecular dynamics, that's great. It's amazing. But sometimes it gets difficult in matching what we're seeing experimentally to what we can see computationally.
Mm-hmm.
And sometimes building up from a more mesoscale model where we don't need to go fully atomic, we can make some assumptions. It gives us really nice feedback that matches what we see from experiment.
Very true. So we'll jump into the questions here. So the first one is, what are the risks of freezing base pairs that you have to watch out for? I think this was kind of going back towards the beginning of the talk.
Oh, that's actually a really great question. I don't know why I'm running into an issue where I can't transition quickly enough in this. So I don't know what's going on. So I want to apologize if I paused.
You're fine. You're good. Yeah, I was having the same issue earlier. I don't know what. I think PowerPoint just doesn't like it sometimes.
Twitch, let me go backwards. Let me go backwards, Twitch. All right, let's go all the way. So let's go to those circles that I mentioned here.
Yeah. Okay. I think this is what
the
question was about. Yeah.
Risks of freezing. I think the risks of freezing, as if you take a look at the circular structure here, you'll notice that based on now the circle that we have here, this is made from a Monte Carlo calculation in which we wanted to look for a Gaussian distribution of circles with specific bends.
Right.
And you'll notice that site 2 and site 4 are really close together in space. So I wouldn't say that this is technically an issue when it comes down to the frozen regions, but so much the frozen regions based on our calculation. Everything is step parameters,
Okay.
which means crossings can occur. So we always have to pay attention to whether or not the frozen regions are too close to each other, where we might run the risk of phasing or a possibility that they're going to overlap in space particularly.
Okay.
In addition, if we were to go through and construct these models here, we showed this example where we can impart one nucleosome, right? Well, I can also do two. And sometimes if I do two and one nucleosome bearing model is really close to its neighbor, we run the issue of overlapping it completely. So that's basically the biggest headache, but that's more about our calculation, basis, the fact that we're doing step parameters, rather than the just the freezing out altogether. Some other things that we have to pay attention to would be with like troubleshooting, in which if we were to create a model and we freeze a region, but we don't freeze anything else, the data becomes useless because we need to maintain constraints all along. If we just constrain the frozen region, allow the DNA to flop around, we don't really get much finding from it. So we have to pay attention on how we constrain on the entire problem. So that's a great question. And I'm glad that you asked that because I haven't had that question yet. So
let's see. How many base pairs can this modeling tool handle?
So I use the term mesoscale, and mesoscale seems to be a buzzword in genomic structures because certain sizes and certain regimes have different terms for it. The way that we define mesoscale here is that I call it's more the binding interaction level. So the amount of DNA in which protein will interact in multiple locations. So the way that we're conducting this calculation, we use a gradient descent method, where if we have an initial condition, we continue to minimize to some standard upon this gradient threshold. Now, certain sizes are going to take a lot longer. So we feel comfortable that at 500 base pairs is that breaking point between reliability and feasibility. I can have a really highly bound structure where I could have like 8 nucleosomes and a bunch of DNA. But if I was to run this on a supercomputing cluster and we ran like with electrostatics, it takes a very long time. So it being 1-to-1, we can even parallelize it all we want to, but we typically want to stay no more than 500 base pairs. I ran some circular work with 1,000 and it took forever. I think it took like 1 week. And that's just because when you have all these factors at play and it being 1-to-1, with that gradient descent method really kind of limits that. But it's a trade-off. But that's the beauty of supercomputers, is that the nice thing about this is that I can run this on blast and get like 100 circles highly robust in like a day, which is great.
Okay. All right. That makes sense. Let's see. Could this type of model be extended to use with polymer chains?
So I'm thinking like polyethylene or or any type of like cellulose.
I'm imagining like anything that has like polymer units. So it could be the same unit repeated, or maybe like 2 different ones. Like I don't know, I can't think of a good example, but like any I guess any repeating polymer unit chain.
Well, here's something really cool. I had a colleague about 4 or 5 years ago who decided, well, the collagen collagen peptide is a triple helix. Well, my colleague, I think it's Faye. So Dr. Xing, what she did was she created base pair step parameters for each item in the polymer, in the collagen polymer. So if there's a level of regularity, If you know the orientation and the position of the regular repeated unit, you can convert that into base pair frame.
Okay.
And then you can optimize the base pair frame. Now, the catch is going to be when you want to create the actual modeling itself, because our calculation is simply in base pair parameter space. So you have a polymer, you know where your origin is going to be located, you know how you want to label the orientation of that polymer in space. Great, you run the calculation. Now you've got to figure out a way to go from base pair parameter space to a modeling space that your program would be like, oh, if I see this, I turn my polymer into this. So it's possible, but it'll take some programming on your part to do that calculation conversion.
Okay, that's pretty cool.
And that's the nice thing about this approximation method. I didn't have to go full atomic for it. If I know my atoms and I know some level of like regularity, you can make that approximation as long as you feel that that approximation space is consistent. It's why we don't use aDNA, because there's no regularity. aDNA has buckling, so that it's not a rectangle. It's more of a like a cup of some sort. So we can't really model aDNA very well.
I got you. Okay, that makes a lot of sense. Let's see. What further improvements can be made to this model?
Oh, okay. So let me there's a lot we can do here. The one thing that I would really love to try to do myself with this model, there's two things. I want to improve the way that we look at electrostatics in this, because the electrostatic calculation is great. But if you've taken physics, or if you have a master's or like a minor in physics, you know that rectangles and electrostatics do not mix, because end constraints or like edge effects, it makes things more confusing. So if you ever took a look at a lot of calculations for electrostatics with DNA, they treat everything like a point charge, which is fine. You could treat it like a sphere. So it'd be really nice for me to go through and try to enhance how we can apply electrostatics, because right now we're putting a charge on the central base, the base pair origin, and then we're also making sure that no base pairs can phase through after a certain size. The other thing I'd like to do is, hopefully in the next few years, I'm not a software developer. I am a mathy nerd person, and I'm using this tool to do my work. But if I were to go to develop anything, I'd really like to optimize on the second order approximation, meaning that we can start optimizing with respect to base pair parameters. I had mentioned that we have 6 base pair step parameters, how one base pair is oriented relative to its neighbor. But each base pair has its own set of 6 parameters. So it would be really cool if we can say, all right, well, we've optimized in base pair parameter space. Now let's optimize to step parameter space to see, or vice versa, where, okay, we've optimized steps, and now let's incorporate some buckling or propeller twisting to see how those differences in base pair A versus B, how those changes can influence the optimized structure. So that's where I'd like to go, as well as some, new modeling techniques or new modeling ideas, like easily developing a circle. And by the way, one thing I didn't mention with this particular project, if this modeling software is something that you might be interested in and you're not really a good programmer, I didn't know anything about computer programming when I started my dissertation. So utilizing this emDNA software, it was intimidating. So one of the things I wanted to do personally was that I published a paper in the Computational Methods Journal, special issue for the Journal of Molecular Biology this year, that showcases emDNA. But rather than just talk about how great the software is, I included a series of tutorials. So if you want to try utilizing emDNA on your own, we talk a little bit about how you make an initial condition, how you optimize it, how you can work with the command line tool with Linux operating systems and work with Python scripting. So if you are out there listening and you want to play around with this as a tool for your own lab, you can do one of two things. One of being, go check out the Journal of Molecular Biology. I'm the first author on that paper. So you can look up young, 2022, whatever, or you can feel free to email me, and I'm always happy to offer some feedback or collaborative experiences on what we can do to get you comfortable with utilizing this tool for your own modeling software, for your own modeling, work pipeline.
That's fantastic. I'll make sure to include a link. I'll look that paper up and include a link to it on the description of this video. That'll be a very handy thing. So the last question that we have here, is there any benefit to going beyond tetrameric context, for instance, like maybe hexameric is next?
That is a great question. And one of the things that we had noticed from our collaborator was they collected data in terms of the hexamer. And that's great. But it actually, the reason we're choosing the tetramer is that experimental biologists have actually said for a long time, the dimer is great, AA, AT, all of that, that's great. But technically, the true subunit for a DNA sequence should be the tetramer, because the internal dimer is going to be influenced by what's before and after it.
Okay.
Going anything beyond that, there are too many monomer subunits at play to really find out what's really influencing curvature. So tetramer, don't think of the tetramer as 4 bases. Think of it as an internal dimer with neighbors. Now, I'm sure you're probably rolling your eyes thinking, well, that's literally a tetramer. But all of our calculations are still at the dimer level. But our rest state, our rest state value is based on what the sequence of the tetramer is. If we do hexamer, there's too many factors at play for us to really feel comfortable with our approximation. But I'm really glad that you asked that. It's a really great question to ask in terms of what the best context is.
Okay, so I get, so in practice it doesn't work. It doesn't work right now for the reasons you stated. But if somebody was really insistent on trying to do it, they would have to basically update their assumptions and approximations. Is that what I'm getting?
Pretty much. So let me, if I was to go to the slide that I discussed, the like tetrameric model in general. Keep going. Here we go. Okay. So the sequence alone is 256 sequences. Now, you might have been some people might have been confused about why we have unique. And what's nice is that with our base pair step parameters, certain tetramers look identical from coding strand to complementary strand. So AAAA tetramers would actually look identical to a TTTT tetramer. Because AAAA in the complement is TTTT in the coding strand. So we can make the assumption that if we see on the coding strand AAAA or TTTT, we can collapse that into one tetramer. Now imagine how much bigger that you got to deal with for the hexamer. That's going to be 4 raised to the 6th, which is 4,096 sequence steps that you got to factor in. Not to mention uniqueness and everything, it starts to get a little unwieldy.
Okay. That actually makes a lot of sense. That's a really good explanation of that. Fantastic. I think that's all the questions that we have. So I will say thank you again, Robert. That said, I mean, this is fascinating work, and you did a really nice job explaining, how you got from start to finish on the work. So thank you for sharing the work with us. And if I can get you to hang around for just 1 second or just a minute, we'll talk very briefly after. And yeah, thank you again. I really appreciate you agreeing to give this talk.
I'm so happy to be here. Happy Science Palooza, everyone. Thanks for joining. And I just find me on Twitter, find me on LinkedIn or whatever. I hope you had a great time, and hopefully get a chance to talk more about this later. Thanks, everyone.